really want to thank the organizers for inviting me to share some of my work under this huge umbrella of Precision Medicine 2019. I'm sure we'll be listening to many experts in the technology development part. So what I'm going to do in my talk is to focus on the applications of the next generation sequencing technologies in identifying the drivers of some of the most aggressive cancers, particularly breast cancer, pancreatic cancer, and prostate cancer. So before I begin to go into the identification of driver genes of aggressive diseases, I would like to mention a little bit about the Precision Medicine initiative that was launched by the National Institute of Health in 2016 with approximately $130 million that were allocated to the National Institutes of Health, and about $70 million were allocated to NCI. And the initiative was described as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle of each person. And the overall aim of this initiative was to understand how a person's genetics, environment, and lifestyle can help determine the best approach to prevent or treat a given disease. So with that, we would like to get into what exactly the contribution of next generation sequencing technology has been in the precision medicine. So precision medicine actually offers a lot of advantage over the traditional cloning-based capillary sequencing in terms of high throughput, in terms of simultaneous monitoring of diverse components of biology, including the gene expression, alternative splicing, chimeric read-through transcripts, and many, many more. And based on this, these advantages, there are many platforms that were developed to understand the genome or the transcriptome in greater detail. And some of these technologies I have highlighted here, including Illumina sequencing, Thermo Fisher Scientific, Pacific Biosciences, and Nanopore sequencing being the leader in the market. And although the platforms of these sequencing technologies, principles are different, but they overall serve as one single goal. And the goal is to obtain maximum possible information in minimum possible cost. And not surprisingly, next generation sequencing technology has emerged and evolved so fast that now we have been able to bring down the cost of the genome sequencing or transcriptome sequencing in a very, very drastic manner. So the question is, how can we use the NGS technologies in the precision medicine in diseases? So in our lab, we study cancers, in particular, as I said, breast cancer, pancreatic cancer, and prostate cancer. And what I'm going to do is to give you two examples of how the NGS technologies have enabled us to find out drivers of these diseases. And I'll be talking about two approaches that our lab has taken in the recent past. One is the sample-centric approach, and one is the gene-centric approach, as I've highlighted here. So a sample-centric approach is where we look at a particular gene, when we look at a particular sample and we ask, what are the genes, or what is a gene that is deregulated in this particular sample? And can we identify the function of that particular gene, and can we therapeutically target that particular gene? So as the name suggests, it is a very sample-specific thing, sample-specific approach. And one of the examples of this the success of this particular approach has been a report that was published in New York Times 2012, where a patient with ALL was treated with sunitinib, targeting the FLT3 kinase that was identified to be deregulated by RNA sequencing while the whole genome sequencing in the same patient had failed to identify any driver mutation or any actionable mutation in this particular case. But as the name suggests, this is a n equals 1 case, so it may not fit all. And above all, it does not consider the inter-individual heterogeneity of a patient population. The gene-centric approach, on the other hand, is when we ask what is a gene or what is a set of genes that is or are deregulated in across the population. So this kind of approach is better with long-term when you have the data, clinical data available for the long-term clinical follow-up. It is also advantageous in the fact that it also addresses the inter-individual heterogeneity of a disease. And so this way it can help identify the genes that may serve as therapeutic targets across the disease site in a population at large. But whatever the approach is, the goal is simple. The goal is to identify actionable aberrations and match those aberrations in patients with available therapies, available targeted therapies. And when we speak about targeted therapies, kinases are one of the most actionable class of therapeutic molecules. And we have seen many success stories of different kinase inhibitors in clinics across the disease sites. And so in our lab, we are focused on identifying the driver kinases across the disease states because they are easily therapeutically targetable. So I'm going to talk about I'm going to start with the sample-centric approaches in cancer first, and then I'm going to talk a little bit on the gene-centric approaches in identifying the driver genes. So what we did is, first of all, to develop a centralized or a generalized matrix to identify the driver genes, in this particular case, the kinases, across the disease spectrum. We thought to use the next generation sequencing, particularly RNA sequencing, on about 482 samples from 25 different tissue types. And we looked at the entire human kinome, that is 468 kinases, across all these samples. And here I'm highlighting only the breast cancer samples, in which we look the kinase for the kinases with extreme expression, and we name these kinases as outlier kinases. And as you can see here, that we've used 43 cell lines and 67 tissues for the breast cancer. And we looked for outlier kinases in each of these samples. And you can see that, as is highlighted by these arrows, that we have identified sample-specific kinases that have extreme expression in different tissue types. And in the inset, you can also see that there is one particular sample, that is BT474 cell line, and we have identified a kinase with extreme expression, that's ERB2. And so the question is, how did we define our outlier expression of the kinase? So we defined it in terms of the absolute expression of a particular kinase in that particular sample versus the differential expression, which is defined by the median level of expression of that particular kinase across all the samples. So we set up a cutoff in this particular case that we will look for the kinases that are at least 5-fold high or 5-fold high differential expression and at least 20 RPKM of absolute expression. And we, as is highlighted in the inset, sample-specific kinases were identified. So the next question is, what is the functional relevance or therapeutic relevance of these outlier kinases? So to assess that, what we did was we used the cell lines and we knocked down their respective outlier kinases using specific siRNA. And as you can see here, for example, in the top panel, the ZR7530 cell line that expresses high levels of ERB2 when we knock down ERB2 using specific siRNAs, we get a drastic reduction in their proliferation. While another gene that is not an outlier expression, RPKS, when we knock down that, we don't see any difference in the proliferation, suggesting that this particular sample is addicted to this particular kinase, and this particular kinase, ERB2, in this sample can be used as a therapeutic target. A similar example is on the bottom panel, which is MDA-MB453 cell line. And here we identified two outlier kinases. One is FGFR4 and one is ERB2. And on the right-hand side graph, you can see when we knock down the FGFR4 or ERB2 or both in combination using either a small molecule inhibitor or specific siRNA, we can get significant reduction in the proliferation. And this is very important because in the clinics, we have time and again seen that the resistance to a given therapy emerges inevitably. So with that in mind, what we thought is we should see whether if the resistance to one of these kinase inhibitors develops, can we use the other kinase as a therapeutic target? And so with that in mind, what we did was we generated a trastuzumab's resistant cell line of MDA-MB453. As in the previous slide, we saw that this particular cell line expresses high levels of ERB2. So we generated a trastuzumab resistant cell line, and you can see that this particular cell line is on the bottom left panel. You can see that this is a trastuzumab resistant cell line that does not respond to trastuzumab at all, but it still responds to FGFR4 inhibition using a PD compound. And BT474 cells here are used as a negative control because they do not express FGFR4. So this data suggests that if you have more than one outlier kinases in a given sample, and if the sample develops a resistance to one kind of a kinase inhibitor, you can still use the second kinase, outlier kinase, as a therapeutic target in this particular sample. And so this gives you a lot of room to use either combination therapy or intensified therapies. So with this matrix in mind, we thought, what about cancers that are highly aggressive, such as pancreatic cancer, that is actually the fourth leading cause of cancer-related death in the United States? So we use this same matrix to identify sample-specific outlier kinases in about 22 pancreatic cancer cell lines and 13 tissues. And again, just like breast cancer, we identified sample-specific kinases that have very high expression in respective samples. And as is shown in the inset, the ASPC1 cell line expresses extremely high levels of MET kinase. And so we can use the MET kinase as a therapeutic target in this particular sample. So as a proof of concept, what we did was we again took the outlier kinases in a sample-specific manner, and we used specific siRNAs to downregulate those kinases. Here, for example, on the upper panel, you can see the L3.3 cell line, which expresses very high level of EGFR, responds very nicely to EGFR knockdown using specific siRNAs in the middle panel. While the AXL gene, AXL kinase, which is not an outlier kinase in this particular sample, does not provide any growth advantage or disadvantage. Similarly, on the bottom panel, the BXPC3 cell line, which has very high expression of MET, and if we knock down MET using specific siRNAs, we get a drastic reduction in the proliferation rate, but any other kinase that is not heavily expressed, it does not make any difference to the proliferation. Now, pancreatic cancer is characterized by or driven by KRAS mutations. So we thought to test whether there are cell lines that are dependent on KRAS and whether we can use the outlier kinase as a combination therapy in with KRAS. And so what we did was we took MIAPACA2 cells, that is a KRAS dependent cell line. It has a mutation in KRAS, and that's why it's highly aggressive. And in the background of KRAS, we inhibited or we knocked down PLK2 using a small molecule inhibitor. So in the left panel of MIAPACA2, you can see that PLK2 is an outlier kinase in MIAPACA2. In the middle panel, you see the shRNAs, that is red fluorescence tagged, that we infected the shRNAs for the KRAS, and we have a very almost all the population expressing the shRNA for the KRAS. And so, and in the right panel, you see that combined targeting of KRAS and the PLK2 offers a significant disadvantage in growth rate of MIAPACA2 cells. In the bottom panel, on the other hand, we have a KRAS independent cell line, which is PANC1, and you can see that neither the knockdown of KRAS nor the inhibition of PLK2 makes any gives it any growth disadvantage. And so this is a proof of concept study that if we identify an outlier kinase in a given sample, we can use that as a therapeutic target in that particular sample. And finally, we tested the impact of inhibition of an outlier kinase in a given sample in the xenograft models. So here I'm showing you a cell line called PANC1. We grew the xenograft and treated the xenograft using a AKT inhibitor XL184. PANC1 expresses very high levels of AKT, and as you can see that AKT inhibition in this particular xenograft resulted in a drastic reduction in tumor growth. And on the bottom right, you can see a Western blot showing that the small molecule inhibitor has very effectively reduced the AKT levels in this xenograft. So to summarize this part of my talk, we can say that we have successfully developed a generalizable matrix to define and prioritize personalized target spectra specific to individual tumors. And this is a proof of concept study that outlier kinases can be used as effective therapeutic targets, either alone or in combination with the known drivers of a disease, for example, KRAS in pancreatic cancer. And so this study was a step forward to the precision medicine in a truly personalized form. So the next question is, what happens when a disease is not as heterogeneous, is much heterogeneous compared to breast cancer or pancreatic cancer? For example, prostate cancer, which is known for its heterogeneity and is known to be a multi-step process. So typically what happens is when a patient is presented in the clinic with a rise in PSA level, he is treated with some form of a localized treatment, for example, radiation therapy, followed by which he experiences a decline in a biomarker called prostate-specific antigen, or PSA. So what happens eventually is that he experiences the rise in this PSA, and at this point of time, he's given the first-line anti-androgen therapies because prostate cancer is driven primarily by the androgen receptor. And this therapy is called ADT, or androgen deprivation therapy. So after this ADT, usually the PSA level drops down, which is an indication of a responsiveness. But eventually and inevitably, most of these cancers develop to a disease state called the castration-resistant prostate cancer. And this is characterized by, again, a rise in the PSA level. And at this point of time, patients are offered a second generation anti-androgen therapies, which is enzalutamide or abiraterone, which are systemic therapies. And then what happens is these therapies definitely give a 3 to 4 months advantage, survival advantage, but they are not of curative intent. And eventually the patient progresses to a very advanced and lethal prostate cancer. And so given a multi-step process of prostate cancer and given the heterogeneity in prostate cancer, we thought that it would be ideal to use a gene-centric approach to identify new therapeutic targets apart from androgen and see whether those therapeutic targets can be of. So to do that, what we did was we took a cohort of patients which is characterized by high-risk prostate cancer, we collected the radical prostatectomy samples from these patients, which we obtained from Mayo Clinic. There are 545 patients in this particular cohort. And the good thing about this cohort is that not only we have the expression data of each of these patients, but we also happen to have a very long-term clinical follow-up data of these patients, which ranges usually between 10 to 13 years. And so with this kind of data, what we can do is we can establish associations between the gene expression data and the clinical outcome data. So we divided this cohort into two groups. The first group is patients that experienced metastasis after radical prostatectomy. And the second group is patients that did not experience metastasis or progress to metastasis after radical prostatectomy. And we looked at the kinases in each of these two groups and ranked the kinases based on their fold change in expression, as you can see here in this particular graph. So on the y-axis, you have kinases that are ranked by their fold change in expression. And we identified a DNA-dependent protein kinase, or a DNA PK, as the top differentially expressed kinase in patients that subsequently progressed to metastasis. This is highlighted here specifically with the red bar. So the question is, what does the high expression of DNA PK mean in terms of clinical benefit? So we looked for the prognostic impact of DNA PK in our other cohorts. So as you can see here in the discovery cohort, that is the Mayo Clinic cohort of 545 patients, high expression of DNA PK is associated with poor metastasis-free survival. We also validated our data, our observation in a separate cohort of 355 patients obtained from Johns Hopkins. And again, the same observation that the high expression of DNA PK is associated with poor clinical outcomes. Now, just like the breast cancer, prostate cancer is also categorized in the basal and the luminal subtype, with basal is known where the basal is known for as a less AR-driven prostate cancer, and the luminal is known as the more AR-driven prostate cancer. So we asked, what is the prognostic impact of DNA PK in each of these subtypes of prostate cancer? And as you can see here in this graph, whether the basal or the luminal DNA PK is prognostic, and the high DNA PK expression is associated with poor metastasis-free survival in these subtypes of cancer. And finally, we also looked for the publicly available databases. We looked at the expression of DNA PK in Oncomine, and we found that DNA PK is enriched into metastatic samples compared to the primary in 10 out of 12 cohorts available. And so taken together, this data indicates that DNA PK is highly prognostic in prostate cancer and is highly associated with metastatic progression. So the next question we asked is, what does the high expression of DNA PK mean in terms of its functional significance? So we inhibited a DNA PK using a laboratory grade compound called NU7441, or we used specific siRNAs that targeted DNA PK in 3 different cell lines. C4-2b C4-2b is a castration-resistant prostate cancer cell line. So is LINCAP-AR, and the PC3 is an AR-independent prostate cancer cell line. And as you can see, that the inhibition of DNA PK resulted in drastic reduction of the in vitro migration assay, in vitro migration of these cell lines. So we wanted to ask, what is it that DNA PK doing? What pathways DNA PK is regulating? So what we did was we performed a guilt by association analysis of DNA PK related pathways, both in the cell line models and in the clinical samples. And we asked, what are the common pathways in the cell line model and in the clinical samples that are associated with DNA PK? In this graph on the left-hand side, you can see on the x-axis, we have the clinical samples, and on the y-axis, we have cell lines in which we knock down DNA PK, and we put them together and ask the common pathways that are regulated by DNA PK. And as you can see, the Wnt signaling pathway emerged as the top pathway that is regulated, that is associated with DNA-PK. In fact, when we checked for the Wnt pathway members in the cell lines that represent the disease progression, where LINCAP is a hormone-sensitive cell line, and C42B and LINCAP-AR are the castration-resistant prostate cancer cell lines, we see an increasingly high expression of the Wnt genes in these cell lines, suggesting that the Wnt pathway is contributing towards the development of the CRPC. The emergence of Wnt pathway has gained a lot of attraction in very recent past. There are studies that have shown that as soon as you start treating patients with localized ADT or second generation ADT, the Wnt pathway emerges. There are whole exome studies and transcriptome sequencing studies that have shown the deregulation of Wnt pathway members in metastatic prostate cancer samples. In fact, when we treated LINCAP cells, which are hormone naive cells, with ADT, that is, we grew them in charcoal strip media, which gets rid of androgens in the medium, we saw that they start to grow slowly. As is seen in the left-hand graph, the gray line shows the LINCAP cells that are grown in charcoal strip medium. They grow slow, but eventually they pick up. And just at the time when they were picking up growth, we looked at the Wnt pathway genes at this particular time. And as you can see on the right-hand graph, the gray bars denote the elevated expression of Wnt genes in these ADT-resistant cells. And when we knock down DNA-PK in these ADT-resistant cells, you can see that the expression of all the Wnt genes denoted by the red bar come down to the normal level. And in fact, when we measure their growth rates, DNA-PK knockdown in these ADT-resistant LINCAP cells drastically reduces their proliferation rate, indicating that DNA-PK indeed is regulating Wnt signaling. So this is about the LINCAP cell, which is a hormone-naive cell. What about the castration-resistant cell line? We looked at the LINCAP-AR cells and also at the C42B cells, which are the CRPC cell lines, and we downregulated DNA-PK either using a siRNA, or we inhibited DNA-PK using a small molecule inhibitor NU7441. And you can see that both at the mRNA and at the protein level, the Wnt genes go down. So the next question we asked is whether DNA-PK inhibition can abrogate Wnt-induced cancer phenotype. So as you can see here in these migration assays on the left-hand side, when we induce LINCAP-AR C42B cells with the Wnt3A ligand to induce the Wnt signaling, they get more aggressive. But when we inhibit the DNA-PK in this Wnt3A induced state, they stop migrating, indicating that DNA-PK inhibition is impactful in abrogating the Wnt-induced cancer phenotypes. So, as I explained in the prostate cancer progression earlier, that after the ADT, after the first line ADT, patients are given the second line ADT, which is enzalutamide and abiraterone. So we asked whether DNA-PK inhibition is impactful in enzalutamide treated or enzalutamide resistant cell lines. And as you can see here, that the LINCAP-AR cells that were grown for enzalutamide resistance, they also respond very nicely to the inhibition of DNA-PK using two different compounds, NU7441, which is a laboratory grade compound, and CC115, which is a clinical grade compound that is now in phase 1 or phase 2 clinical trial. So the question is, if DNA-PK is regulating Wnt signaling, how does it doing it? So mechanistically, we show that DNA-PK actually interacts with the Wnt transcription factor LEF1. And as you can see here in these co-IP graphs, when we pull down DNA-PK, we are able to pull down LEF1. and vice versa. We also show using a reporter that reads the transcriptional activity of LEF1, that DNA PK is required for the transcriptional activity of LEF1. So in the right-hand graph, if you see when we induce the cells with Wnt3a, the reporter activity goes up. But when we inhibit the DNA PK using NU7441, the reporter activity drastically goes down, indicating that DNA PK is transcriptionally required for the LEF1 function. We also performed the chromatin immunoprecipitation studies and noticed that DNA PK is enriched onto the regulatory regions of Wnt genes. And that's how it regulates the transcriptional activity of LEF1. And when we inhibit DNA PK using NU7441, we see a drastic reduction in this enrichment of DNA PK on the Wnt genes. We finally tested the impact of DNA PK inhibition in vivo. Here we use two different xenograft models, LinCap-AR, which is a CRPC xenograft model, and PC3, which is an AR-independent xenograft model. And we used DNA PK inhibition using two different compounds, either NU7441 or CC115. And in the leftmost panel, you can see that LinCap-AR xenografts respond very well to DNA PK inhibition by NU7441. In the middle panel, we see that DNA PK inhibitor CC115 performs much better than enzalutamide in reducing the growth of the xenografts. In fact, when we give CC115 in combination with enzalutamide, we see a much better reduction in the xenograft growth. In the right-hand side, rightmost panel, we see a PC3 xenograft, which is an AR-independent model of prostate cancer. That even in this model, DNA PK inhibition is very impactful in reducing the xenograft growth. So to summarize this part, we can say that the gene-centric approach, using this gene-centric approach in prostate cancer, we identified DNA PK as the top expressed kinase associated with metastatic progression of prostate cancer. We also show you the data that inhibition of DNA PK suppresses the growth of both the AR-dependent and AR-independent prostate cancer cells, and that it does so by regulating or inhibiting the Wnt signaling pathway. So with this encouraging data, we are testing the DNA PK inhibitor CC115 in a phase 1 or phase 2 clinical trial in metastatic CRPC patients in combination with enzalutamide. So what are we looking forward to with these kind of gene-centric or sample-centric approaches that are enabled by next-generation sequencing technology? We are looking at the platforms like whole genome sequencing and transcriptome sequencing, and eventually that can actually both of these sequencing forms can now identify different forms of oncogene activation, different structural variations, as we have published the whole genome sequencing of 101 metastatic castration-resistant prostate cancer patient. But what we are looking forward to now is to use integrative approaches where we can integrate the whole genome sequencing data with the transcriptome data and start to see what are the deregulations in these kind of cancers, whether these deregulations can be therapeutically targetable. And so this slide pretty much summarizes what I just said, that integrative omics approaches can lead us to achieve maximum clinical benefit. So we can take a patient's tumor and we can use different approaches that are enabled by next generation sequencing technology. For example, genomics or epigenomics or even immune profiling or transcriptomics. And then we can integrate this data to look for robust biomarkers. We can validate this data to look for the potential drivers. We can do the network analysis data. We can develop the metrics to properly stratify the patients into proper risk groups that can be benefited by special therapies or specific therapies. we can look for the small molecule inhibitors or immunotherapies or various kinds of combination therapies with the goal of achieving the maximum possible benefit to the patient, good clinical outcomes, and rationally designed clinical trials. And so, with that said, that next generation sequencing technologies have enabled us for a lot, but there still remains many challenges for the precision medicine initiative and the use of NGS in this particular realm. First of all, we need to really standardize the collection of clinical and hospital data centers, because this initiative is spread across the centers in the United States. We need to be able to design databases to store the large amount of sequencing data coming from whole genome sequencing or transcriptome sequencing in a very efficient manner. We have to also find ways to protect the participants' privacy and the confidentiality of their health information. We certainly need to develop a rigorous process of informed consent. And eventually, we need to employ the practices that help us in lowering down the cost so that the technology is available to more population in general. And finally, in my own opinion, we need a better understanding of these technologies through interdisciplinary approaches. We need basic scientists, we need bioinformaticians, and we need medical professionals to come together, join hands, and brainstorm to identify the right kind of drivers for the right kind of disease, for the right kind of therapies. With this said, I believe that the future of the next generation sequencing technologies in precision medicine look bright. As we say that a picture is worth 1,000 words, so we definitely are unzipping a lot of dark clouds in this area, and we are looking to a bright future of next generation sequencing technology and their application to the precision medicine. So with that, I would like to acknowledge a big team that has helped us in these kind of studies. And I definitely would like to acknowledge our funding sources, the Prostate Cancer Foundation and the Department of Defense, for funding our research. And I'm happy to take any questions on this presentation. And I welcome all of you to ask your questions via emails. I assure you that I'll be replying them very promptly. Thank you very much.